Our next topic of discussion is for IB B2.2, and this is focused on organelles and compartmentalization, really looking at how are different organelles adapted to have specific functions that help the cell to be able to survive and carry out its function. Organelles are identifiable structures in cells that are adapted to perform specific vital functions. They are specialized for that specific function. And there's many different types that are found in eukaryotes. Sometimes they can be single or double membraned. And then also some have no membrane. So those that have no membranes would be ribosomes, centrioles, microtubules, proteasomes, and the nucleoli. Single membranes would be vesicles and vacuoles, the rough endoplasmic reticulum, and then also smooth endoplasmic reticulum, Golgi apparatus, and lysosomes. And then lastly, organelles that have double membrane would include a nuclei, the mitochondria, the chloroplast, amyloplast, and chromoplast. And so those different types of organelles can have different membrane structures to be able to create an internal and an external environment. There are some components of cells that are actually not organelles, and this would include the cell wall. Because it's outside of the plasma membrane and extracellular space, it's not actually an organelle itself. The cytoskeleton is made of protein filaments, and it has limited separation from the other structures within the cell, so it's not an organelle. And then lastly, the cytoplasm, which includes other components with different functions, and so it's not itself a discrete structure within the cell, though very important to the survival of the cell. Prokaryotes, in comparison to eukaryotes, have much less organelles, fewer organelles. They're smaller, and it's a more specific range of functions. And prokaryotes don't have any membrane-bound organelles. They do have ribosomes, obviously. They have DNA, but the prokaryotes don't have those generalized structures that we would think of as organelles. So we'll start by looking at the nucleus and examining the specific functions and adaptations of different organelles. And the nucleus's job is to keep the DNA safe. And a nucleus is found in eukaryotic cells, and the purpose of it is to provide a protective bubble, if you will, really to be able to ensure that the DNA isn't damaged, because DNA is used to be able to make proteins, provides the instructions to make proteins. It's essential that that DNA remains the same and is not damaged, and that's the purpose of the nucleus. Prokaryotes don't have a nucleus. The DNA is just clumped together in a nucleoid region, and so within prokaryotes, translation, the second step in the process of making proteins, it takes place immediately after transcription, the first portion in the process of making proteins. And so it's just one right after the other, because the messenger RNA that's made in transcription doesn't have to leave a nucleus. It's all right within the cytoplasm. Within eukaryotes, because there's the nucleus, that messenger RNA is made during transcription, and it must move from the nucleus to the cytoplasm. And in that process, it can be altered before translation takes place, and this results in a variation of the different proteins that can be produced. So that's actually kind of an advantage. It provides greater variation. And so the function, the purpose of the nucleus is to provide protection for that, for the DNA molecule. Because organelles have membranes, it creates a compartmentalization that provides some advantages. And these would include the ability of enzymes and substrates to be more concentrated inside of a contained area. It helps to keep harmful substances out or in, depending on the situation. For example, digestive enzymes contained within lysosomes so that they're not seeping out into the cell. It's contained within that lysosome, containing or maintaining pH levels for a specific process. The ability to move specific contents within the cell or around the cell or outside of the cell through vesicles. And it also can increase area on a membrane for reactions to take place. And then a good example of this that is essential is in the mitochondria with the production of ATP. So it's actually really beneficial for most organelles to have membranes to be able to create some compartmentalization. Speaking of mitochondria, the mitochondria is an organelle that has a lot of unique characteristics and features, adaptations that help it to be able to perform its function, producing ATP. And so to start, it has an outer membrane that separates the biochemical reactions that take place of aerobic respiration. It also has an inner membrane, and that inner membrane contains the proteins and enzymes that are used in the electron transport chain, including ATP synthase, that are used to be able to produce proton gradients and create ATP. Part of that, because of this inner membrane, it creates this structure called cristae, which are tubular projections, and they increase the surface area that's present, really just allowing for more proteins to be embedded within this inner membrane and more space for the electron transport chain and thus ATP production. There's a space between the inner and the outer membrane. It's a pretty small space, and it allows for the easy creation of a concentration gradient for hydrogen ions. And those hydrogen ions, as we'll see a little bit later, are used in the electron transport chain to produce ATP, are essential to that production. And then also, lastly, the matrix inside of the mitochondria is kind of like the cytoplasm of a cell. And it contains the enzymes that are necessary for the Krebs cycle, a portion of this process of aerobic respiration, as well as the link reaction. And by being confined to this area, it increases the rates of reaction for both of these reactions. A second organelle that has some similarities to mitochondria would be the chloroplast. And this organelle is adapted to be able to use sunlight to create energy, specifically sugar. It's got some similarities to mitochondria, first being that it has a double membrane, and inside of the chloroplast itself, it has small, kind of like disc-shaped structures called thylakoids. They have a membrane themselves, and they're green due to the presence of chlorophyll, which reflects the green light. Inside of the thylakoid are small fluid-filled spaces. The fluid itself around the thylakoid is called the stroma, and this, similar to the matrix or to the cytoplasm, is where enzymes are contained in order to be able to carry out reactions. Oftentimes, chloroplasts have grana, which are stacks of thylakoids stuck together. So if you imagine an individual thylakoid like a coin, a whole stack of coins would be in the grana. The purpose of the chloroplast is to absorb light, and pigment molecules within the photosystems of the thylakoid membranes are absorbing that light. It's got a large area of membrane or a large surface area to ensure lots of light absorption. That's essential to being able to produce the sugar. ATP is produced, as we'll see, in the process of photosynthesis that takes place in chloroplast. This is produced through photophosphorylation. There's a hydrogen proton gradient that develops inside and outside of the thylakoids, and that thylakoid volume itself is very small, thus to be able to create the gradient. We'll look at this process of photosynthesis a little bit more in the future, just like aerobic respiration. The chemical reactions of the Calvin cycle take place within the stroma, and it produces some ATP and a molecule called NADPH, and those are required or necessary in order for photosynthesis to be able to take place and produce sugar. And so the chloroplast, much like the mitochondria, has some specific adaptations and structures to be able to perform its function of, in this case, producing. The nucleus itself actually has a double membrane, and that membrane is made of phospholipids. And because the phospholipid bilayer has a hydrophobic core of membrane, if there are holes that develop in the membrane, they always are pulled back together because of these the hydrophobic protection of the hydrophobic tails. And so this results in shapes without edges, spheres, in order to protect that hydrophobic core. The membrane of the nucleus must have pores or openings so that different substances can move in and out, proteins, messenger RNA. Those pores are essential to allow specific molecules to pass through, but they're generally small, as larger holes would result in some catastrophic damage and would thereby damage the DNA. And so the pores themselves within the membrane of the nucleus are formed by using integral proteins. And this allows proteins to regulate gene expression, or it allows messenger RNA to move in or out of the cell, as well as ribosomes. Larger than normal pores are necessary, and so a double membrane enables a larger pore lined with protein to control whether or not a protein can traverse, can move in or out. That membrane can reform after breaking during mitosis and meiosis due to it being phospholipids, and the arrangement or protection of the hydrophobic core. And so during that process of mitosis and meiosis, the nucleus membrane actually dissolves, it breaks apart, it can reform because it's made of phospholipids. The nuclear membrane breaks off into vesicles, spheres, that can later reform into a new membrane by fusing, as we previously discussed with the cell membrane. Ribosomes, although not a membrane-bound organelle, are essential to cells because they perform the function of making proteins. They synthesize proteins, and ribosomes are made of rRNA, ribosomal RNA, as well as proteins themselves, and it's in two different parts, a large subunit and a small subunit. The large subunit has binding sites for a molecule called T, or transfer RNA, and the small subunit is what the messenger, or mRNA, actually binds to. Free ribosomes are synthesized polypeptides for use within the cytoplasm or in the nucleus of the cell. Attached ribosomes, so those that are attached to the endoplasmic reticulum, thus making it rough endoplasmic reticulum, synthesize polypeptides proteins that are transported to specific locations, usually the Golgi apparatus, which are oftentimes secreted outside of the cell through vesicles that bud off or branch off from the endoplasmic reticulum. The Golgi apparatus is an organelle that's closely tied to the endoplasmic reticulum and the production of proteins, and it is essentially a stack of flattened sacs called cisternae that process the proteins produced by the endoplasmic reticulum. Those proteins are transported by vesicles, and enzymes in the Golgi apparatus can change the protein, the polypeptide, by adding carbohydrates, phosphate, sulfate, establish quaternary structure. It's really where the proteins are being modified for their final use. After that processing, they're transported by vesicle to the destination, sometimes within the cell, oftentimes outside of the cell, and that the movement of those vesicles is sequential, in which each protein gradually moves through the cisternae from the side closest to the endoplasmic reticulum to the opposite side, and so it's moving through the Golgi apparatus. There's two models of how this movement takes place. There's not a lot known about the Golgi apparatus, so we're not exactly sure which model is followed as the correct model. One would be the vesicle transport model, in which the cisternae do not move and the vesicles just do the transferring. The other is the cisternal maturation model, in which the vesicles from the endoplasmic reticulum combine to form new cisternae that then move to the opposite side. And so one of these two models is probably how this process takes place, but we're not entirely sure which is the correct method. More evidence and research is needed. Our last organelle to look at are vesicles, and these are rounded sacs made up of a single layer of membrane. They're small, they're continuously made, they're moved around and merged within cells by pinching off small areas. And how that process works is through the use of a protein called clathrin. It's a three-legged structure that positions on the inner face of the plasma membrane when the vesicle is being made. And adjacent molecules then bind to one another to form a support structure, and it allows the plasma membrane then to become indented and eventually detach or pinch off to form a sphere with clathrin around it, thereby forming the vesicle. And typically vesicles are used for one of two primary different functions: movement of contents or movement of membrane. A good example of the movement of contents, something within the vesicle, would be the transport of neurotransmitters to presynaptic membrane of the neuron, so actually moving molecules around. The second method would be the movement of the membrane itself. This would be like to increase the size of the plasma membrane after secretion of membrane proteins within the vesicle of the membrane. So replacing a portion of the membrane that's lost, or adding to it in order for the cell membrane to be able to grow in size. And so this concludes our looking at specific organelles and their functions or adaptations to be able to help them and help the cell in order to survive and carry out its functions.